So the entrance of a carbohydrate, it's going to begin with either glucose or glycogen, and that's what we talked about with glycolysis. When oxygen is present in large enough quantities, the end product will be pyruvate and gets sent off into Krebs cycle, or also known as the citric acid cycle. And then we'll look at a pictorial in a second to see what happens. So remember that this is taking place inside of the mitochondria, the organelle, inside of the cell itself, and we have a few places where we can enter. As you see, amino acids can enter as pyruvate or oxaloacetate or fumarate. Fatty acids can be converted into acetyl-CoA, and if you remember that glucose or glycogen is converted to acetyl-CoA after it goes through glycolysis, ends with pyruvate, gets shipped off into the mitochondria, and that conversion of pyruvate to acetyl-CoA will take place. Similar to the reactions that we see in glycolysis, multiple chemical reactions that spin off either ATP or some other constituents, and most notably are going to be hydrogen ions here. So this is the electron transport chain. Again, also taking place inside of the mitochondria. You see we have the dropping off of the hydrogen ions. So it's the use of the hydrogen ions that turn these things called cytochromes, and we phosphorylate ADPs to produce ATPs. Incidentally, this is the greatest amount of ATP production that will take place of all the systems. So all the hydrogen ions get to kick off and are picked up by either NAD carriers or FAD carriers from glycolysis or from Krebs cycle, and they're sent off to the electron transport chain in the presence of oxygen to be converted into, eventually, carbon dioxide or water are going to be the byproducts of aerobic metabolism. CO2 is off of Krebs cycle, and water is going to be a byproduct of this process. So when we use glucose or glycogen as the original substrate, that takes place in glycolysis. So long as oxygen is present, then we get to take the pyruvate, enter the mitochondria. Also, those hydrogen ions carried by NADs will also enter the mitochondria. The pyruvate will be converted into acetyl-CoA to spin Krebs cycle, which also kicks off even more hydrogen ions. And the hydrogen ions from fast glycolysis, or slow glycolysis we should say, will move to the electron transport system to produce an oxidative phosphorylation. So for each molecule of glucose, we actually get to turn Krebs cycle 2 times. Now, lipids, fatty oxidation, is going to take place by the breakdown of triglycerides, and that hormone is called hormone-sensitive lipase, which is an enzyme that converts the free fatty acids that allows it to go through beta oxidation. So through beta oxidation, we take the carbon molecule chains that are further broken down into acetyl-CoA and hydrogen, and they can enter Krebs cycle and the electron transport system. So these are only performed, this process is only going to be oxidative. There's no glycolysis for this or something similar. It takes place during oxidative metabolism. We don't think of protein oxidation as a major source of energy when it comes to muscle contraction and the use of ATP. However, especially during either starvation events or very high and intense long duration activities, we can break down amino acids, convert them to glucose, pyruvate, and enter Krebs cycle. We use branched chain amino acids, leucine, isoleucine, valine are the major amino acids that are used. So you may have actually seen supplementation of these for energy production. The deamination process is where we remove that amino group, the nitrogen complex, and that's the production of urea. So we don't like to use protein as an energy source simply because of its byproduct and therefore the metabolism. So this is just another slide showing all the different energy yielding nutrients and protein, carbohydrates, and lipids or triglycerides, just to show that they can all be part of the oxidative system and essentially where they enter the process. So there's an inverse relationship between these systems as to how quickly they can supply the ATP and their amount. The quicker that they're available to supply ATP, essentially the lower the amount. So the phosphagen system, it runs out very, very rapidly because it has a very, very low amount compared to the other systems. Our most efficient system is going to be the oxidative system. It essentially can supply us with a limitless supply. However, it takes a very, very long time to get there. So for really intense activities, we need it more quickly. For less intense activities, such as being at rest, we don't need it as quickly. So our most efficient energy system is going to be the oxidative system. So there's also a relationship between efficiency. Although one has greater capability, we also need to make sure that we can supply the demand. So this is looking at a complete yield. Remember that we said with glycolysis, if it ended in the cytosol, in the cytoplasm there, then our yield is going to be very, very low. So we don't produce a great amount. However, if oxygen is present in large enough quantities, you see that most of the ATP from slow glycolysis actually takes place due to the oxidative system. So the substrate phosphorylation itself, glycolysis without the oxygen piece, it only gives us 4 ATP. However, if we can transfer into the mitochondria, you see that the total yield, because of the oxidative phosphorylation and substrate phosphorylation of acetyl-CoA from pyruvate, we actually get a total of 40. So much, much better at producing total quantity in presence of oxygen. Now we look at one triglyceride molecule, and we'll assume that the triglyceride is made up of 3 fatty acids that have 18 carbon molecules. Glycerol itself, the backbone of a triglyceride, gives us 22 molecules of ATP, and then the 18 carbon fatty acids times 3 gives us a total of 441. So a total of the 3 molecules of fatty acids and 1 molecule of glycerol, we get a total yield of 463 ATP. So in comparison to 1 molecule of glucose, the yield is much, much higher. Therefore, in terms of overall supply in numbers, the phosphorylative system oxidatively is going to be much more efficient in the overall numbers. Again, remember that it's going to be slower.